thumbs up. All right.
Yeah.
All right, great. Well, welcome back everybody for day 3 of our virtual BioGeoscapes US Planning Workshop. We appreciate you coming back to join us for the 3rd day. And today, Naomi and I will be mostly your hosts. So we wanted to start with another acknowledgement, one that I know the organizing committee has been feeling, and I bet many of you have as well, which is the challenge of being in a virtual meeting, particularly one that lasts for a couple of hours each day over multiple days. We had really hoped to hold this meeting in person, and obviously weren't able to, but we just want to acknowledge the challenge of sustaining engagement with a virtual meeting. Often when you're at home or when you're dealing with life issues at home, and you're not sequestered in the beautiful conference room in Clark. So thank you everybody for making the effort to be here. We really appreciate the ideas, the energy, the thoughts, the presentations, the posters, and the time that everybody has contributed to this meeting. Okay, so here's our plan for the final day. We're going to start by trying to summarize the breakout discussions from yesterday and the Jamboards that came from that. We're then going to have 4 more talks by different speakers about sort of potential examples of this type of science that at least the organizers have thought could be really exciting to undertake at a larger scale through a BioGeoscapes program. We're then going to have a breakout discussion today and report back from that today, since we don't have another day to mull those thoughts over before we get back together. Then we're going to spend a bit of time talking or hearing from Mike Saraki from NSF about sort of how we should be thinking as a community about trying to build this kind of program. And then we'll have some time at the end, hopefully a fair amount of time. It won't feel quite as rushed as some of our other moments have felt so far to sort of wrap up, look back on what we've agreed upon and sort of communicated about here and talk about next steps as a community. Okay, so I'm going to start, Naomi and I are going to sort of alternate trying to summarize the 3 questions that were posed to each of the breakout groups yesterday. And for each of these, we have a sort of a summary Jamboard, and the sort of charge to the group is in the upper right. So here we said, what hypotheses could be addressed by a BioGeoscapes program toward the goal of coalescing around high-level motivations? And in reading through all of the sort of the answers and ideas that were put forth for this, we've tried to sort of summarize these in a couple of ways. Some of these are, maybe are sort of slightly more mechanistic ideas or questions. And then I've sort of at the top level, I've tried to think about the sort of high level motivations that would drive those questions. So for example, a common idea was the really high level question of what controls the fate or the persistence of organic matter in the ocean. So clearly that's got a lot of ramifications for carbon, for productivity, for the life cycles of all of these things in the ocean and their biogeochemistry. And some of the ideas that fell below that, or I sort of grouped with that, were, for example, anoxic processes, both in low oxygen parts of the water column, but in oxic water column regions, where I think there's increasing recognition that those might be important to biogeochemistry. What controls phytoplankton loss rates? Obviously, a really key question about the fate of organic matter. Bunch of different ways, probably more ways than we often think about for where phytoplankton can be lost, and how can we study that? A number of groups asked or mentioned, what are the drivers, just the drivers of autotrophy and heterotrophy in the ocean? How can we understand the controls on those levels of activity, on the size of those communities, on the diversity of those communities? And also, what about mixotrophy? Again, an area that has gotten much less study over the past decades, but I think has increasing, or at least a lot of questions about its importance. There were a number of groups that talked about ocean primary production, or NPP, and how will that change with time or with climate conditions. And sort of related to that, there was sort of a general sort of topic about using omics to diagnose things that are not as easy to measure directly or over the same scales that you might be able to do omics. For example, nutrient stress, or viral pressure, or grazing pressure. Processes that we know are important ecologically, biogeochemically, but are hard to measure and hard to measure in a standardized way. And maybe omics can provide a really useful tool for that. A related topic, similar maybe, but slightly different than the other two, what controls transformations and vertical fluxes of organic carbon in the ocean? Right, so not just the production of it, but all of the transformations, including what happens to it at the end. And for example, what is the role of nutrients and micronutrients in structuring the microbial communities in the ocean, as well as their function? And then in the lower right, I tried to summarize a number of sticky notes that sort of talked about the importance, particularly for models and for prediction, of understanding the relationship between environmental conditions, community and function, diversity and function, and then the resulting rates. I think in some part of me says, well, of course, that's what we should be doing. But I think it's really valuable to point it out that these are key relationships that we still really struggle to generalize to the global ocean. All right, Naomi, on to you.
So the second question focused on thinking about the power of a biogeoscapes type research program, specifically the power of collaborative interdisciplinary research and the research that requires a large scale coordinated program. And there were a lot of really great ideas that emerged from this discussion with some reoccurring themes that again, we tried to summarize here. For example, several of the groups identified that science that aims to link processes across scales really can only be done through these types of collaborative work. Similarly, questions aimed at quantifying scales of variability and those that need to sample over different time scales are all examples of research that would be challenging without a coordinated program such as biogeoscapes. In terms of the types of science that can be addressed, several groups pointed to global spatiotemporal variability of biogeochemical transformations that organisms and understanding the organisms that drive them, which is a bit of a mouthful, but I think it actually is a really succinct capturing of what a lot of these biogeoscapes type questions are and how we need these interdisciplinary groups to address them. The kind of similar frame is studies that are linking rates and community structure and understanding who is where and what environmental factors are determining these communities and their functions are all other examples of questions that really can only be addressed by these types of coordinated efforts. And finally, there was some comments linking highlighting the power of integrating this work through modeling, especially metabolic models, and the power of cross-disciplinary work that brings together experimental, observational, and modeling groups, and again, how coordinated programs such as biogeoscapes would really facilitate this. And provided that we really think carefully about the communities that we're bringing in, there was this comment that we should think about linking in the eDNA community.
Great, thanks. Okay, so the third charge for the breakout discussions was to think about what are the key intercalibration needs to help support and realize our science goals. And some of these we talked a bit about, or we gave some feedback on in the Mentimeter yesterday. On the left, I'd say, there was certainly conversation in a number of groups about, just what is the best way to intercalibrate complex analytes. It's not, although GeoTraces probably provides, in some cases, a good model for how to approach it, in terms of a structure, the actual process of figuring out, are we measuring the same thing or are we making comparable measurements is much more complicated with many of these omics analytes. And so I think that's still something that we need to figure out. There was also some flavor of discussion about how much omics intercal is necessary. Not is it necessary, but how far do we need to take it? How far, how close do the measurements need to be? What is practical, what is feasible, and what is necessary to answer those science questions from the previous slides? And similarly, sort of as Naomi alluded to in the previous Jamboard, there was some discussion about, what is the role of modeling? Can modeling inform the intercalibration needs in terms of maybe looking at the size of responses that we would need to sort of ground truth with these measurements? There was discussion about the specific types of analytes that are needed to intercalibrate. Certainly, there was rates came up. That was the winner in the Mentimeter poll yesterday. Discussion about chemical omics additionally, and in particular metabolomics. There was discussion about things like FlowCam and flow cytometry, sort of often pretty basic or other complementary ways to characterize biological communities that, generally we don't check to see if we are getting sort of comparable answers with those approaches. Certainly nucleic acids got a lot of discussion, and there's a lot of effort around that and will happen around that. And then some other examples that through no choice, but just we're given a small font here, eDNA and the issues of standardizing that, lipids, metabolites, microscopy and other imaging, ways to measure biology and fixation rates and primary production. So those are the ones that really got multiple points of discussion. In pink, you can see the discussion around the need to standardize or intercalibrate collection and processing. Obviously, time scale is a really important factor there. Filters sort of approaches is always one that deserves discussion, and also archiving. The acknowledgement that these technologies are changing, and I'm sure will continue to change, and the potential value of having samples from the past that can be analyzed maybe with future techniques in order to provide a bridge as technologies change. Funding and the need for funding, the recognition that these intercalibration exercises are complicated and expensive and require significant effort was recognized by a number of groups, as well as the potential to learn and make closer connections to other communities, for example, NIST or NIH or maybe others. And finally, there was discussion about the value of sort of the cookbook, one that has pretty detailed protocols, and acknowledgement that in the case of these bioinformatic techniques, the access to pipelines and the open sharing of pipelines and standardized bioinformatics behind those pipelines was also really important, and something that a group like GeoTraces probably didn't have to address with such importance. And finally, the availability of databases got some discussion, as well as ways to standardize measurements, for example, through mock communities or other, again, broadly available tools that for running and standardizing your data. Yeah, I think that's it. Great. So, we will have a bunch of opportunities in the discussions later today and at the end of the day for feedback on those, for sure. So, if you really think we've missed something there, please speak up later when we give you those opportunities. And also you can always email and communicate with us other ways. Okay, so we're going to move now to sort of introducing today's speakers. And although Naomi practiced more than I did for pronouncing Daniela's name, I'm going to give all of their names here. So our first speaker will be Daniele Aiuticone from Italy, and then Lahini Aluhare, Sarah Hu, and John Casey. And Naomi, take it away.
All right, Daniele, do you want to share your screen for us? So it's my great pleasure. All right, good. So it's my great pleasure to introduce Daniele Aiuticone, who is coming to us from Italy. And thank you so much for spending the Friday evening with us. We're really excited to hear about Atlantico and other things you tell us about.
So, hi everyone. Thank you for inviting me to present our project. And I will start saying that it's nice to see that the items you are discussing are precisely the same we are discussing here in Europe in different committees and things. So it would be really great to keep a cooperation on those points, on all those points, scientific and technical ones. So let me present quickly. Our project is a European project that is dedicated to assess the status of the Atlantic ecosystem health and ecosystem and provision. And it's a huge project. It's 11 million, 5 years, and 36 partners, with 5 from Brazil and 3 from South Africa. The general aim is to have a new holistic framework for the management of the ecosystems and to engage with the science, industry, policy, and citizens, and to build a shared capacity. But we are going to focus specifically on the microbiome, on the Atlantic microbiome, and on the plastic and the plastisphere, so microplastics and plastisphere, within the context of the seascape. So generally speaking, the challenge is to put together the 3 context system. One is a seascape, which is with a very specific properties that we cannot extend terrestrial theories to that. The complexity of the organism and the biology of the organism, together with the complexity of the interactions, the interactome, as we say now. So here, this is, if you want, the challenge we share. And in our project, we try to focus on different pieces of the story and trying to connect the pieces sometimes when possible. The general idea is to have a circular approach, an holistic approach, where we go from the all measurement, all data to new, collecting to new data, but always connecting with labs, with bioinformatics, data science, modeling, etc., trying to have a dialogue. This regard, so we build the project into different work packages, but basically the activity streams are four. One is to assess the status of the ecosystem. And in this regard, I think it would be interesting for you to know that we are collecting all the global data and genomic data and trying to reprocess them with a unique pipeline. So that we will soon release a first database that can be used by everyone, where all the data from entire oceans to the recently released Ghost Ship data are going to be together and homogenized. There will be work on drivers with some socioeconomists, and there will be a lot of new observations that I actually will describe a little bit in detail later on, some theoretical analysis, and then work on capacity building, communication, outreach. So I want to detail a little bit what we are going to do in terms of sampling. We have a series of flagship quizzes, which are going to be empowered with the Atlantico kit, which is an evolution of the Tara Oceans protocols that I will illustrate later on, but basically it's adding omics, so genomics, transcriptomics, metagenomics, and metabarcoding using a standardized approach and a size fractionated approach. We are going to analyze the past data by the INT, for instance, in the Atlantic, but also empower the next expeditions. Then we are going to work with our Brazilians and South African colleagues for cruises. We had then a series of sailing boats going around. The first and most important I will discuss in a minute with the new expedition with the Tara vessel, but then there is also a German boat, and then there is a Brazilian boat that are going to be use the same protocols and going around in the basin in the next years. The Tara Ocean Expedition, the new Tara expedition, which is called Mission Microbiomes, is kind of our flagship, which in a sense that it collects not only the omics data protocols we somehow know, but it's trying to push forward and introducing also trace elements. Together with Ted Jones, I would say, in the USA, and eDNA, isotopes, long reads instead of short reads. I mean, together with short reads to try to compare, advanced microscopy, metaproteomics and metabolomics, where metaproteomics is with Max Saito, Guzzo, and then microplastic. We are going to split this expedition. We are splitting this expedition, which started this summer, into a series of topic studies, so process studies on different subjects, from the impact of iceberg on the biome, on the biota, to main rivers, to the south, the Weddell Sea, thermal circulation, to the Amazon plume, and seamounts impact and the mesoscale effects. As an example of process studies, so process studies are organized in terms of there is a dedicated team which includes colleagues from the neighboring country. It is dedicated to a women from that country, a researcher from that country, so they all have a name that is actually the name of researcher of Brazil, Argentina, South Africa, etc. And is directed by young people, so we decided to have chairs, the young, and always respecting a good gender balance. And what I want to tell you in this case is in Patagonia, is that from tomorrow we're going to start this joint experiment with another sailing boat, where we're going to just share the sampling and the protocols. And what is nice is that the sampling boat is the Atlantis. The former Atlantis from Woodsole, which is an historical boat, and now is owned by the Argentinian Navy. And so we are empowering now it's called OSE with our protocols, and we are going to sample together in the area in sort of historical encounter. I mean, something I really like personally as an oceanographer. Then we are going to analyze also sediment traps with omics, and also empower a new CPR line in a region where there basically were no data. So there is a new CPR line that just started between Dubai and Rio. And we are going to have also simplified protocols to be used by marine stations around the basin, And also we have a citizen science program, which even simpler approaches that will be really shared at all possible scales. We are developing, as most of you know, probably an handbook of standard and best practices, where basically we have the list of all the protocols, something we have been organizing with the workshop last year with some colleagues also from US. And I think this is something we should talk in the future, because this could become a shared knowledge or shared use, and we could develop this together in next years. And there is a special issue on these topics in Frontiers, just started. We have a hub and nodes approach, so we are also empowering our Brazilian and South Africans with sharing not only capacity and tools, but also connecting the sequencing centers, trying to have a shared standards also the sequencing level. So some of the temples are going to be sequenced locally. And then for the impact, we have multidisciplinary training, summer schools, and we have a dedicated program with UNESCO on literacy, on the microbiome, the ocean microbiome, and then we have a series of case studies, applied case studies. I want to finish this quick and fast, sorry about that, overview with some general comments on what I can maybe bring to this workshop. So one thing is that in addition to the Atlantico, which is going to produce a lot of data, and I want to in the handbook, but I want to underline here that we never had the money to sequence all the data. So if you have an interest in sequencing some of our data for your studies, you're completely welcome. There is within Bioscapes, we are also developing an observatory here, which is implementing Atara Ocean routinely since 3 years. So we have monthly data, and also here we will be happy to share information and collaborate. But then I think it's important politically, we have two news. One is that the European level, the EMBRC, which is an infrastructure for biology, European level has started the EMOBON, which is a coordinated sampling effort monthly in many marine stations, so using omics in Europe. And I think that would be, and they have an handbook, which is a sort of simplified subset of our protocols, and we are coordinating on that. And then there is a new EuroGoose, which is the European flavor of goose, that is a working group dedicated to biological ops. And there too, we will work on the coordination at European level for protocol standards and anything that relates on it. At global scale, I think it's really important that you're aware that there is one of the framework, the project approved by the UNDCA is actually dedicated to molecular observation, so the oceans, it's called OBON. And I think this is a perfect place to empower Bioscapes. So I think I'm in the steering committee, and I think it will be really important that we connect the two programs. And this is even more important because in the G7 working group where I am since so many years, there is now a new interest in empowering this kind of new augmented observations. And so this could be the place where we have a political support to then ask for money at country level, of course, but that's something we should consider. So I'd be happy to be in contact on all these issues, and I'm in all these committees, and so that's it. Thank you very much.
Great. Thank you, Daniele. That was great. Such a great reminder of how much is happening outside of the US. Which is really important. Okay, we're going to go on to the next talk, and we'll have questions and comments after all 4 of them. So the next speaker is Lahini Aluahare, who is a professor at Scripps Institution of Oceanography. And Lahini, do you want to share your screen and take it away? We can see the presenter view, not the full screen view.
Thank you. All right, is that better?
Yep, perfect.
Thank you. Okay, so I'd like to start off by thanking the organizers for giving me the opportunity to speak here today. I've been spending the last 25 years or so of my career thinking about dissolved organic matter cycling in the ocean, and more recently I've been thinking about how I could leverage programs like Bioscapes to provide some insight into that discussion. So a lot of what we know about DOM cycling in the ocean has really been provided by concentration measurements across the different ocean basins, and thanks to folks like Dennis Hansel and Craig Carlson, we have really good coverage of DOC concentration measurements. And those concentration gradients have allowed us to glean some information about the potential rates of cycling of different DOM reservoirs. And from the context of, in the context of biogeoscapes, perhaps labile DOM, which facilitates the very fast cycling of carbon and nutrients, and semi-labile DOM, which actually stores carbon and nutrients on decadal to multi-decadal timescales, might be most relevant for this program. In the context of labile DOM, many folks in this room are interested in the potential for secondary metabolites that are part of the DOM reservoir to structure organism-organism interactions, and we're interested in this as well. And from the perspective of semi-labile DOM, one of the things that I've been interested in, many of us have been interested in for some time, is the unusual, unexpected accumulation of organic carbon and nitrogen that we see in Jaya region. So this is DOC concentration data from P16 compared to the radiocarbon signature of DOC. And we see that the most enriched radiocarbon signatures are in these locations where we have accumulations, suggesting that this material is labile, but potentially on decadal or subdecadal timescales. And we don't know why these accumulations exist in the ocean. And I choose these two examples because sort of the exometabolite or non-targeted approach that we take in our lab to look at and to examine these interactions between organisms and metabolites really benefits from being embedded in a program that measures a variety of different biogeochemical parameters so that we can really interpret those exometabolite profiles in a meaningful way. Whereas sort of this broader scale question about the accumulation of labile DOM in gyre systems may actually benefit from sort of a more survey type approach where targeted omics type measurements may be leveraged to study why we see these differences in the accumulation. So from the perspective of microbes and metabolites, we've been with our collaborators collecting various field samples for exometabolite work from different locations around the world. This sample set looks at compounds that were extracted in exactly the same way, analyzed on the same instrument, and put through the exact same pipeline, all of which is available publicly. And you can look at a very broad scale relationship using something like principal component analysis at the compositional relationship between these different environments. And you can provide this very dizzying array of a molecular inventory of, in this case, 56,000 features from 2,800 samples. Unfortunately, with the available library data sets, we can only annotate just over 11% of these molecules, which leaves about 90% of the molecules in these data sets with little or no structural information. And so an effort in my lab spearheaded by Irina Koster, who's a student with me and who's somewhere out in the Zoom ether, is really trying to work on how to provide more relevant chemical information for the dark metabolome. And you can track her down and talk to her more about, talk to her more about that. But you know, what's the point of generating these sort of molecular inventories? We can't understand what these molecular fingerprints mean from an ecosystem context. So to address that kind of a question, I've been focusing a lot of my exometabolome work in the California Current Ecosystem, LTER, where a variety of different biogeochemical parameters are measured. And so here's an example of a satellite chlorophyll image from June of 2016. And the framework for these studies is that we conduct Lagrangian experiments that last 3 to 4 days and look at the ecosystem parameters and how they evolve over these cycles. We measure a variety of different hydrographic, we collect hydrographic data to allow us to assess the integrity of the Lagrangian scheme, as well as look at potential connectivity between these different cycles. For example, this study was designed to look at how an upwelling filament evolved in its chemistry and biology as it moved offshore. So we can use the hydrographic data to look at where water moss upwelling originated and how it evolved and moved across these environments in this filament and look at how organic matter might be transformed as these filaments move offshore. And of course, we collect a variety of different chemical measurements as well here. We're looking at how DOC and nitrate are related at these different locations in the study site. Of particular importance is that we also quantify carbon fluxes and exchanges between different compartments of the food web. And so we can even get sort of really broad scale estimates of trophic state in these different environments. So for example, we know the coastal region in this system was net autotrophic, which means there's excess DOC production, for example, that's available to be exported and transformed as it moves offshore in the filament. So looking at things like vector production measurements can give you an index for the extent to which DOM is being transformed or accessed. And looking at depth profiles of things like ammonia or how the isotopes of suspended PON change with depth might allow you to isolate locations where there's intense reworking of organic matter and try to connect our metabolite data in the context of those processes. In addition, we're lucky that we have a variety of different investigators associated with this. So for example, we know that we can quantify viral infection rates or copepod fitness, and using some of the tools that Bethany Edwards talked about yesterday, we can actually use metabolites to give some context for the processes that are taking place in these environments. Or we also have information on ion limitation of phytoplankton and bacteria, and we can see how metabolites might give us a picture of ion limitation at some of these locations. We also can collect, of course, RNA measurements, and we have the ability to do transcriptomics, but what we've been focusing on lately right now is really just community composition. And based on this data, we can see, for example, that these high productive cycles are enriched in organisms that are known to degrade complex carbohydrates and proteins. And that's consistent with what we see with this excess DOC production and high bacterial production. In offshore environments, we see that the food web, of course, is based in Prochlorococcus production, and that might be relevant in terms of how we interpret metabolites. Whereas where we see ammonia production, for example, we see that there's a relative increase in the abundance of sequences related to nitrosamine cumulus. So this sets the context for some of the interpretation that we can engage in when we look at these exometabolite data sets. So for example, we can look at the fingerprint of metabolites as this filament moves offshore, and using sort of a very coarse principal component analysis context, and it can tell us something about how these different water mosses may be related. So we see where a diatom dominated production environment is in cycle 1 is quite unique relative to some of the more closely related water mosses that are evolving as we move offshore. And so while we can try to interpret the metabolites in this context, and an easy thing to start with is sort of looking at correlations between microbes and metabolites to give a sense of sort of this interactome that we've been discussing. So in cycle 2, where we see the iron limitation, for example, we detect a fungal metabolite, diacetoxyskerpinol, that's correlated with the presence of a fungus based on the 18S data, as well as other heterotrophs. And so while the deck board incubations can tell us something about iron limitation and alteration of the metabolome from a phytoplankton perspective, the metabolites can give us some sense that maybe there's the potential for fungal siderophore production in this location, for example. And then when we move offshore, where we see the Prochlorococcus-based food web, we see strong connections to heterotrophs that we know are typical of these oligotroph environments. And those interactions are mediated through a variety of different metabolites, including some that are highlighted here that are signaling molecule and manoylide, a toxin that's known to be produced by cyanobacteria that's associated with sponges, for example, so likely originating from Prochlorococcus, but we of course have to ground truth that with more targeted studies. of particular interest to me, and this is a bit of a complicated figure, but I'm just going to summarize it quickly, is that as these water masses are moving offshore, we can also look at how organic molecules might be transformed, not just oxidized to CO2, but their chemistry altered. And so by looking at compounds that are related, both in terms of their composition and also in terms of their changes in abundance over the course of this filament, we can try to identify mass changes or mass differences that are most frequently observed. And I'm interested in this because I think that this is a pathway by which semi-labile DOM may be formed in the marine environment. And so bringing that back to our discussion about semi-labile DOM, we talked about the accumulations of DOC that we see in these gyre environments, and those are accompanied by regions where there's really very little nitrate or no detectable nitrate, and yet we see accumulation of organic carbon. Chlorophyll is also low. And if you look at TDN, we have excess total dissolved nitrogen that suggests that DON is accumulating in these environments as well. So in these oligotrophic nutrient limited environments, we have the accumulation of carbon, organic carbon and nitrogen. And what is driving this, right? So there's a variety of different processes that might be important, but the potential for sort of sequential alteration of these organic compounds such that they become less palatable to the communities that live in the surface ocean may be a question that we can leverage using something like biogeoscapes with very focused measurements. So for example, we could, in addition to just measuring inventories, we can also start to ask, are there really simple assays to look at DON lability from a compositional perspective, including DON isotopes, and then combining that with a variety of different omics measurements that can target specific questions, such as, are there changes in the types of enzymes that are being expressed in these different environments. And so just to end, I'd like to point out that Daniel Petrus, who was a postdoc in my lab and who's at the University of Tübingen now, is collaborating with Jeff Hawks at Uppsala to conduct actually a very simple kind of cross-calibration effort for metabolomics data. So it's very simple. We collected, we provided about 20 labs with a particular sample set, and you'll see that in a second. They collected the data on their instruments and they sent the data back. Daniel and Jeff have been analyzing that with their own, with a single data processing pipeline. And, we see what we might expect from the first data sets that have returned, that each lab sort of differs in terms of the global metabolome. But when you compare the most abundant 1,000 or 100 molecules, you do start to see a consensus appear. So this just describes the types of organic matter that we concocted in the lab, including pure DOM. This shows sort of the principal component analysis that shows the differences between the labs for the types of samples analyzed and just the consensus that we see in some of these data sets. So I'm going to stop there and be happy to take questions later. Thank you.
Thank you so much, Lahini. That was fantastic. Great example of biogeoscapes like science. And so we're going to move straight on to the next talk. Again, please put questions in the chat because we will have hopefully a little bit of time for Q&A at the end. So it's my pleasure to introduce Sarah Hu, who's coming to us from Hui. Sorry, can you share screen? Looks great.
Okay. Hello. Thank you for the introduction and the invitation. Today, I'm going to talk about some of the ways I look at the marine food web and work to parse apart or isolate the different trophic interactions. And I'm going to talk primarily about like single-celled microbial eukaryotes in the environment, right? Because they play a lot of different roles in marine food webs. Phytoplankton, photosynthesizers, consumers of other prey, symbiotic hosts, parasites. And this makes it really fun to study microbial eukaryotes, but also really challenging to tease apart those diverse trophic strategies. So I'm going to talk about a couple of ways in which we do that. First, talking about my current work, which is all set at deep sea hydrothermal vents and looking at the role of microbial eukaryotic grazers. And then I'll talk about some outstanding questions, and I have a little example that talks about forward-thinking thoughts on metaomics and how to incorporate that. So, right, we've talked a lot about this meta barcoding and tag sequencing approaches that give us this taxonomic identity. And a key part of that is having environmental context with these snapshots of all the taxa that you're getting in the community. And that's great. It comes with, that's great to leverage things like time series stations, Station Aloha, Spot Station, BATS, CalCoffee. And today I'll talk about my environmental context being the hydrothermal vent and the deep sea. I'll also mention metatranscriptomics a little bit later, where we're looking at the eukaryotic messenger RNA. And I like to think about it as, in addition to getting your environmental context, when you do in situ metatranscriptomics, you get the taxonomic identity and functional potential. And this is literally a way to triangulate and pull out those in situ trophic modes. So to address one of my main questions, I'm looking at these deep sea hydrothermal vent environments, right? This is, I'll show data from these two sites, Gorder Ridge in the Northeast Pacific and Mid-Cayman Rise in the Caribbean. And this is an environment with no sunlight. It's supported by these chemosynthetic bacteria and archaea that are interacting with that vent fluid emanating from the subsurface and interacting with the seawater. And one of the ways I've essentially isolated heterotrophy, or work to isolate and focus on heterotrophy, is removing the sun from the equation. A lot of my omics signatures in the photic zone, right, are kind of swamped by phytoplankton, but to study heterotrophy, I decided to move to the vent environment. Another thing is I kind of think about this as an environmental gradient to explore, which is from the gradient existing from the site of that diffuse flow to the plume and then to the background deep sea water. And this is really exciting to study, because this environment in the deep ocean is this like hub of biological activity. And so what's happening with the food web outside of this hub, within it, and kind of an influenced region, and then right at the site of that hub of biological activity. So to isolate the role of heterotrophy in particular, I ran these grazing experiments. And so here we have the labeled prey that I prepared in the lab next to the what the resident microbial community looks like. And after collecting fluid from these vents, I incubated this and then collected samples over time to then measure the uptake or loss of that labeled prey. And that will allow me to actually quantify grazing. And to add in a qualitative component, I've also done a lot of sequencing of these sites, both the in situ and the incubations themselves. This is going to be a compilation of all of my grazing rates. Along the y-axis is the cells consumed per mil per hour. And here, and this is a log scale as well. Here we have all the background sites from both two vent sites in Mid-Cayman, Picard and Von Dam, and then the Gorder Ridge site. And these are on the orders of hundreds cells consumed per mil per hour. And if we want to look at the hydrothermal vent sites, the grazing rates from the hydrothermal vents, we see a couple of things that are popping up. First, there's a lot of variation in the grazing rates that we get at the site of that diffuse flow interacting with the seawater. And I should mention these are all diffuse flow, low temperature, so less than 100 degrees Celsius. And another take home is that while there's a lot of variation, overall, the grazing rates obtained from the site of the hydrothermal vents are higher than what we see in the background. That's kind of speaking to that hub of biological activity. These things, these microbes are kind of attracted to this environment. There's increased grazing pressure at this site. Another thing is that across different vent ecosystems, we're seeing some values that are in range with one another. So that's kind of exciting. To introduce the qualitative component, which is kind of my main arc here, this is the sequencing data from the in situ samples. So we have relative sequence abundance on this side, and the colors designate the major taxonomic groups. Now, this is all the background signatures, so the community composition. And when we add in the hydrothermal vent environment, we're again reinforcing that gradient that we see from the background to the hydrothermal vent. We're seeing higher relative abundances of things like ciliates, a very well-known heterotroph, so potentially linked to some of this increased grazing activity. I don't have time to go over all the different parameters measured, but this trend from the background to the vent site also corresponds with some trends in temperature, cell concentration, both prokaryotic and eukaryotic, and measures of species richness when I think more about just the presence and absence of ASVs rather than relative abundances. But I want to focus on the quantitative plus qualitative component here, which is, so I'll highlight these two example experiments where we have Shrimpacalypse and Mount Edwards, okay? And what I've done here is show the bar plot sequencing from the in situ on the left and then the incubation itself on the right, with the relative abundance again here. And I'm showing this because by looking at both the in situ and the incubation themselves, we can look for shared ASVs and really hone in on the hydrothermal vent grazer community. Another thing this does is that it's to my advantage in that I now can be a little more confident that the incubation I ran was representative of the in situ environment. So next steps for this include doing in situ experiments. A subset of those were run at in situ pressure, but we have a cruise next year to do more. And then also continue on with doing more metatranscriptomics of these samples. And so I wanted to go into questions, and I'll go off in an offshoot briefly. But one of my main ones is working to connect what's happening in this food web in these punctuated hotspots of biological activity, like a vent system, and then connecting that to the rest of the water column. One of the broader implications of some of this trophic transfer that I'm capturing in the grazing activity of primary produced carbon being consumed by the microbial eukaryotes is that then that's a food source for higher trophic levels. There's going to be export, there's more recycling back into the environment. And that's all kind of in the vicinity of the hydrothermal vent. And some of our calculations suggest that that carbon recycling might outweigh that coming from the euphotic zone right near the hydrothermal vent site. So it makes me want to branch out and kind of connect some of these environments more. Another big question that I think about every day is how do we, again, take apart those different trophic strategies. And I talked about, a qualitative plus quantitative approach with the metabarcoding and grazing, but this could include other imaging, RNA-seq plus metatranscriptomics and cell counts. And one example that we've seen a lot of today, and I've seen a lot of the posters, and I use it a lot, is this ordination approach, right? So this is a example where I've taken different depths from the spot station. I'm looking at the metatranscriptome signatures from 5 of the main taxa. And this ordination, this is the CCA in this case, I'm able to pull out the phytoplankton communities at the surface. I'm able to pull out haptophytes, these mixotrophs. Then I'm leveraging the environmental context of the different depths, the surface sunlit area and then the non-sunlit area, and kind of capturing that heterogeneity that we might see in trophic strategies. And I mentioned that these are always my favorite plots to look at in different papers and all the posters, because you get to see these kind of broad patterns where, like data points, colors and symbols in this case show that there's like a unified nutritional strategy or functional potential or something with specific taxa. And they're, again, leveraging taxonomic identity, functional potential, and often environmental context. So we have the same kind of approach, cultured representatives, mesocosms, and single cell genomics. And this one example here is from a paper that's a preprint where we have over 900 eukaryotic MAGs from the Terra Oceans data. And one outcome from that was assigning a subset of the MAGs to heterotroph or phototroph using the metatranscriptome signatures. And while we know that we have this trophic mode prediction, the heterotroph or phototroph, we know that this is not binary. And so through discussions, we assigned a heterotrophy index. And to me, that's a really advantageous way to approach this kind of work, because you're able to actually embrace the metabolic complexity that these organisms are capable of. And so I want to think about fun ways to effectively apply this to models, qualitatively partitioned boxes. And then my final question is more infrastructure based, where I want to make sure that these pipelines can be run by everyone that wants to learn how to run them, and it's easy to do. And that's going to facilitate better collaboration to pool those resources and references. Thank you.
Great. Thank you, Sarah. That was fantastic. All right, before we go to questions, we have one more speaker, who's Dr. John Casey at MIT. There we go. John's out there. Take it away.
You guys see my screen there?
Yeah, looks great.
Okay. Thanks everybody. I'm really excited to be here and to learn about the vision for biogeoscapes in the early days. I'll basically be kind of beating the same drum today as Scott McCain was yesterday, but from a slightly different angle. So if nothing else, I hope to drive home his point about comprehensive sampling to enable microbial simulations. A common goal here might be to develop prognostic models of the marine ecosystem. So I'm hoping to share some progress toward that goal from systems and computational biology perspective. So I find it's useful to ordinate data with models. So a couple dimensions that we might think about are biological complexity, which is shown on the horizontal axis, and biological diversity shown on the vertical axis. So the contours here are supposed to represent lines of constant compute resources, okay? And the vector field shows the trajectory that computer scientists like to push their models, okay? So domain, time, and space are not represented in this plane. So keep in mind that bridging of scales from molecular to the ecosystem comes with additional trade-offs and challenges. There's two kind of broad categories of approaches that one might take in generating predictions. There's probabilistic or phenomenological models, and there are mechanistic models. So I, like many of you, are just kind of blown away by the power of regression approaches like machine learning and deep learning, but they tend to be difficult to interpret and to learn from. So on the other hand, mechanistic models allow us to simulate processes based upon what we think we know about a system, and then update our assumptions in an iterative fashion. So the most useful models, at least under that reading, would be interpretable at the level of the observations that we're comparing them against. So if we can place our observations within this plane, then the model we choose will depend on how complementary it is in terms of both complexity and diversity. So within biogeoscapes, many of the observations that will be made, you can find kind of in the top right corner of this domain, all the genes, transcripts, proteins, and metabolites that we find in a bottle or on a filter. So this is by no means a comprehensive list, but we might place some broad categories of the kind of current state of the art in modeling approaches that are being implemented in microbial oceanography relative to our observations. So at the broadest and coarsest complexity, we have maximum entropy production models. Then trait-based models like Darwin, which can be embedded in global circulation models, agent or individual-based models, macromolecular models, similar to the ones that Scott described yesterday, and genome scale models. Of course, there are currently no models anywhere near the resolution and scope needed to directly simulate the entire ecosystem at the molecular level, of course. So we need to engage in some kind of dimension reduction in at least one axis. So this dimension reduction can be done either on the data side through, for example, clustering approaches, or it can be done on the model prediction side. So I've been kind of attracted by all the progress in constraint-based modeling and genome scale models. And I see fewer and fewer hurdles toward more diverse microbiome simulations in the not too distant future. So really briefly, genome scale models represent the full metabolic network of an organism as a matrix of the stoichiometric coefficients of all metabolites in all reactions. So the associations between genes, proteins, and reactions are explicit in genome scale models. We can derive a chemical flux balance in an open system from the law of mass action. And if we assume a quasi-steady state wherein there's no net change in the intermediate metabolite concentrations, then we can solve this system using flux balance analysis, which is a constrained linear program which searches for a set of optimal flux distributions which maximize the growth rate of the cell, which in this case is represented by a vector of the composition of biomass. So the optimization is subject to constraints on both the reaction directionality, which we can infer from thermodynamics of the reactions, and from experimental data. So I want to share our first simulations with GEMS in the ocean environment and some prospects, hopefully for future collaborations within biogeoscapes. So the typical FBA problem assumes a static biomass composition, known uptake rates. These are kind of common for bioreactors, industrially relevant microbial simulations. So both of which we know are variable in the real environment. So our framework addresses that problem with a steady state fitness optimization called, which we're calling the microbial simulation environment. Which accounts for acclimation processes associated with not only metabolic reactions, but also with cell size, individual nutrient transporters, all the different pigments, and macromolecular pools. So metabolic and physiological acclimation is simulated in a sequence of constrained bilevel optimizations, which you can read more about. So in environments which depart substantially from their ideal laboratory growth conditions, acclimation can increase growth rates substantially, effectively broadening the niche width, okay? So we have manually reconstructed pangenome scale metabolic networks, pangenome scale metabolic network from 648 Prochlorococcus genomes from isolates, SAGs, and MAGs. And I think the rarefaction curve in the bottom left panel shows that we're getting close to saturating the metabolic orthologs for Prochlorococcus. So we've also developed a pipeline for annotation and with popular bioinformatics, cheminformatics, and systems biology databases and tools, as well as an efficient algorithm for the automated reconstruction of individual strain GEMs, which we've implemented for here for 69 high quality isolate genomes spanning the 5 major ecotypes of Prochlorococcus. So a typical strain, just to give you an idea of scale, will have around 800 genes, 1,500 reactions, and around 1,000 metabolites. So our first application was for an Atlantic meridional transect cruise way back in 2003, which extends from the English Channel to the Falkland Islands. We chose this cruise and sifted through many because of synoptic measurements, not only the physical and chemical fields that are needed to run the simulation, but also for the synoptic measurements of Prochlorococcus ecotype abundances, which are used for a weighted average of all of our predicted rates and stocks for the total population. So the bottom left panel here shows a meridional section of the transect with light levels in the yellow contours, temperature in the white contours, and nitrogen concentrations as a heat map. So both at the population level, shown in panel C, and among the 5 major ecotypes shown in the right panels, E through I, we see a correspondence between predicted growth rates, which is the shading in each panel, and observed abundances, which are the white contours in each panel. The shape of that relation at the population level are shown in panel D for our predictions in black, and the few kind of independent observations that we could find in the literature in red and blue. And they indicate to us that something besides linear mortality, which would have a negative relationship in this plot, is at play. So I show this kind of as an example of how we can infer something about population ecology from the molecular scale. So there's kind of a mountain of data spanning elemental stoichiometries, photosynthetic performance, metabolic rates, cell size spectra, and other quantities, getting down to real nitty gritty that I don't have time for here today, but that I hope you'll be able to read about soon in a forthcoming article in Science Advances. So to kind of get back to the theme of matching models to observations, I'm showing here one example of how we can query the model predictions at different taxonomic and physiological resolutions. So we include a detailed representation of photosynthesis and photoacclimation for each of the strains. And our AMT transect shows a partitioning of the photosynthetic performance and pigmentation between strains, which are the small markers colored by ecotype here, between ecotypes. Those are the larger colored markers, and the abundance weighted average of the full assemblage shown in the black markers. So from these patterns, we can simulate P versus I curves to infer photosynthetic parameters like the assimilation number and EK, which are shown in the right two panels here. And these are quantities we might measure directly in the field. Okay.
Hey, John, how many more slides do you have? Ah, it looks like you're close. Great.
Two more. Yeah, sorry.
Thank you. Great. No problem. Just want to make sure we have some time for questions. Thanks.
Oh, thanks. Sorry. So there, I'll be quick. There are several kind of exciting directions we might go from this first demonstration. We are working on expanding taxonomic coverage to enable community simulations, which I've started on for a number of clades. In terms of dimension reduction, I'm running simulations across a grid of environmental variables to generate lookup tables for Darwin's version of Prochlorococcus as a way to improve and contextualize parameter estimates. And I've also started looking at relaxing the steady state assumption to enable dynamic simulations over the diel cycle. Finally, I'm working on a new acclimation step, which solves the optimal proteome allocation problem, enables us to resolve things like trace metals limitation. So these last two items rely on a capacity constraints on fluxes in the optimization. So I'm working with a group in Sweden on a deep learning algorithm to predict catalytic rates for individual enzymes based on associations between chemical structures, the substrates and products that they interact with, and protein structures, which we're generating with AlphaFold. So I'll cut it off. I must be over time here, and we just want to thank my collaborators. Thanks very much.
Thank you, John. Yeah, no, sorry to just we've packed it all in, but that was great. That was great. Okay, let's see. So I think we have about 10 minutes that we'd love to address some questions for the speakers. I see there's a number of them in the chat. Let's see, Naomi, I don't know if you found, there's also some answers in the chat too, so I don't want to repeat something here, but maybe I see, yeah, go.
Oh, I was gonna say, just to start with the first one, the question for Daniele from Adrian. Do you want to ask that, Adrian?
It actually was answered in the chat. Thanks.
Okay. Oh, yeah, sorry.
How about Xavier's question? Xavier Mayali, Talahini?
The one Talahini, yeah. So, yeah, I was just curious as to whether or not it's as simple in terms of understanding the breakdown of DOM over time, right? Is it as simple as those molecules are going to get smaller because they're being chopped off little by little, or is it actually not at all like that?
Well, it depends on who you talk with. I don't necessarily believe that they're getting smaller per se, because if you think about how, I mean, sure, you might have larger polymers that get broken down into pieces where you know, have bonds that are not as susceptible to breakage given the community or something. But I'm kind of more interested in some of these things like deamination, where you're not just chopping the amine off, you actually are replacing it with something, right? You have to put something in to take something out. And so it's not necessarily that the size is changing. And I think in terms of your other parts of your question, I think, you know, there's no one answer. I think, of course, these culture samples or experiments are really important. And the new STC that's led by Marianne and Liz will really do so much, I think, to address some of those outstanding questions. And of course, transcriptomics, I think, is really helpful too.
Well, now that we have you on the spot here, Lahini, Jesse, do you want to ask your question? Are you there, Jesse? No, Jesse. He was asking about whether you had any thoughts on linking the processes of DOM with specific heterotrophic groups experimentally through isotope probing or such.
Yeah, all of those things. And I think Xavier can answer that question. But yeah, no, I think, I mean, yeah, I don't want to prescribe anything because I'm an old fart. Like, you know, you guys have so many amazing ideas and techniques that all of you are bringing to the table here. And I think there's no one process, but yeah, I think controlled lab experiments that are addressing, you know, we have to validate some of these correlations that we see in the environment. Otherwise people will be really unsatisfied. But that's, you know, we also have like multiple years of data from Cal Coffee's, from CCLTR. So, you know, if those correlations continue to be robust, it'll be hard to ignore them.
I don't want to keep you on the spot, Lahini, but I think Allison's question is also worth addressing, and Kim sort of follows up on it. And others can jump in too, but Lahini, do you have a thought about, you know, how do you approach the small number of identified compounds?
Yeah, I would say this is the one thing that keeps me up at night. Well, actually, no, it's not the one thing, but you know, when I first started, FTIC RMS was really coming online, and there was a lot of pressure for people like me to go after that technique, and I always felt like that I was not interested in that approach because it was really only very good at giving, it's excellent at giving molecular formulas, elemental formulas, but it gives very little information on the secondary structure of the molecules. And then, you know, so I was like, oh, I'm going to do LCMS-MS. And then I come along and I can, you know, identify 11% of the molecules with existing libraries. So we've done a lot. I mean, we're really working hard on using in silico approaches to provide more compositional level information, but it's slow, right? You're not going to do that for 56,000 compounds. And so I think, again, this is where, you know, some of these, looking at some molecular microbe networks and then looking at transcriptomics might actually allow us to get better information on the types of, or more specific information on some of the metabolites that are being produced in these environments. But I am confident that some of these in silico approaches can give us actually like structural motifs and populate some of these networks that don't exist at all in the library. And as I said, my student Irina's been working on this for some time, and she might have some insight as well.
I'm going to move the hot seat from Lahini. Xavier, I really liked your question for John. Do you want to ask it?
Yeah, sure. I mean, John and I have been talking about this off and on in the past too, and I work with some folks who are doing FBA modeling on and trying to use that to investigate interactions. And so I guess my question was, you know, I think FBA is very good at looking at model systems that are well characterized, you know, E. coli and, you know, and Prochlorococcus, I think is the one marine organism that fits really well into that category. And you're doing some fantastic work with it and FBA and looking at this globally, and that's awesome. But can you say something, do you think we can use FBA to look at complex interactions in these ecosystems in its current form, which is my understanding, and I'm not a modeler, and maybe you can correct me and us. My understanding is that it only examines central metabolism, and it does not really have a way to look at secondary metabolite production and signaling molecules and things like that. So can you talk just a little briefly about that?
Yeah, thanks for the question. Great question. And I think Jacob had a kind of a related question about community simulations. So just to clarify, all metabolic reactions that are annotated in the genome are included in the metabolic network. So that includes not only central carbon metabolism and primary synthetic pathways, but also the secondary metabolites are synthesized in these models. Oftentimes they end up being dead end metabolites because there's either no secretion pathway known, or because there's nobody on the other side to take this thing up. So it's not optimal to pass any flux through a pathway like that. Now, if you had a receptor for one of those dead end secondary metabolites in a community simulation, then you would optimally pass flux through that if there was some benefit on the other side. So you get a lot of interesting changes in the flux topology when you include community interactions. So basically, another way of saying that is that growing an organism in isolation in a bottle, you would predict a different metabolic flux than you would if it was in the presence of another organism. And some of that might be controlled over evolutionary time. So there's some kind of suboptimal phenotype that they're just expressing this pathway in the hopes that there's another organism around to give them kickbacks. But yeah, so basically the kind of the model is set up to resolve those kinds of processes. It's just yeah, needs another step basically.
And I'll chime into kind of, in my experience, Xavier, you're totally right, but it's a bias in the data sets and the annotation and our understanding, especially for marine organisms, because a lot of this has been based on E. coli work. And so things like amino acids, we're really good at with the models, especially these like automatic pipelines. And it's not that they can't do these other things, as John was really nicely highlighting, but we need a lot more work in the community to be able to easily incorporate them into these FDA models. And
so sorry, not to keep going on this, but so what do we need to do to improve then those models to incorporate interaction? Do we need more careful experiments with very small communities, or do we need more sort of big data?
So the biggest bottleneck that I can see is transporter annotations. Okay, so this is kind of a common thread. If you wanted to putatively identify interactions between different microorganisms, you wouldn't need model organisms to run careful, detailed simulations. You could identify all these interactions just from transporters. Okay, so what the reality is that there's kind of poor specificity, poor annotations, and just a ton of uncharacterized membrane transporters that you find in just about every genome. So some kind of functional genomics effort would be very much appreciated. Yeah.
He's been chiming in the chat. I agree that we need more lab work, physiological characterization, knockouts, etc. So maybe that can be continued in one of our breakout sections, which we will introduce. I think we're going to move into that now, right? Is that correct?
Yeah, probably should.
All right. So let me share my screen. Just give me 1 second here.
I want to again thank all the speakers. Really great to see the diversity, and really interesting to see the modeling try to now to overlay on some of this massive complexity. It's really impressive.
All right, what are you seeing?
See your presentation view. Looks good.
Okay, looks good.
Yep.
All right, so I think that was a good segue to go into our breakout discussions, which we're going to have and do slightly different than we have done the past 2 days. So today we are going to have a 15-minute breakout with 5 different breakout rooms. However, instead of randomly assigning you to these 5, you will get to choose which of these 5 you would like to go to. So the 5 discussion topics for today, and the numbers here will correlate to the breakout room number, will be for breakout room 1, field measurements, thinking about process creases and time series. And so we're envisioning here that this discussion is going to stay at a higher level rather than getting into the nitty-gritties of how process crews might function. Thinking about more questions such as how can we make this kind of franchise model that we've been kept kind of has come up over the past couple of days, inclusive of a broader community. What are some lessons that we might be able to learn from previous efforts, such as exports or the time series sites or the LTR program, and what kind of science questions could be pursued? So the direction you go will be up to the group, but trying to keep it at kind of a higher level. Similarly, breakout room 2 will be focused on, again, field measurements, thinking about more survey creases and transects. What lessons might be learned from previous programs, GeoChases and Tara, how we might be inclusive again, not just having one group doing all of measurement X. For breakout room 3, we wanted to give some space for the modelers to come together and talk a bit about how we can best build our community, as I'm a modeler, and be active measurers in the Biogeoscapes program. Similarly, breakout room 4 is to try to give some space to these discussions that have emerged and we were just having about how to best include laboratory experiments and culture work. It's come up several times, and so we wanted to present space to expand upon those discussions. And then the last room, or breakout 5, will be focused again on this topic that has come up several times on the desperate need for biogeochemical rate intercalibration. And so we thought we'd have a space to discuss how to best move this forward. So again, unlike previous days, you'll be able to choose which group you want. This slide will come back up in a second. So think about it. So again, just to remind everyone that the goal here is to really maximize the input and hearing from many different voices and perspectives.